Andrea. Good afternoon. It's really a great privilege to be here, so thank you to Andrea and ICRO and the committee for inviting me. Unfortunately, Andrea wouldn't lend me his tie, so I'm tieless here. Actually, as a basic scientist, it's actually, you're very privileged to see me with a shirt and jacket on, never mind a tie. So I'm from Manchester. This is our wonderful new building that we opened last month, and my lab is actually in the building here. And as you can see, Tuscany has the Leaning Tower of Pisa, but we have the Leaning Lab of Rob Clarke in Manchester. And I'm going to tell you today about modeling breast cancer, but also about drug radiation therapy interactions, particularly with CDK4/6 inhibitors. So I don't have any relevant disclosures. So I'm going to tell you something about preclinical modeling of breast cancer, in particular about how to model using patient-derived samples in 3D, in vitro, but also in mice as patient-derived xenografts, where you can also model metastasis. And then in the second part, I'm going to tell you something about CDK4/6 inhibitor mechanism of action, but also mechanisms of resistance, and how there may be opportunities for rational combination with radiotherapy. So this slide here, courtesy of Elisabetta Marangoni, summarizes some of the differences between using in vitro models and in vivo models, both cell lines and patient-derived models, but the differences between 2D and 3D. So you can see in particular, in vitro, you can do high throughput drug screens that you can't do in vivo, but those cell lines in particular are not going to be any good at predicting individual patient responses. For that, you need patient-derived 3D cultures, and in particular, these patient-derived in vivo tumors. And in those in vivo models, obviously, you also have this opportunity to do metastasis modeling, but it's only really with the syngeneic mouse models that you can look at an intact immune system, since when you put patient tumors into a mouse, they have to be immune-deficient mice. So I just wanted to highlight some work from several years ago from my lab, where we did some work on combination of radiotherapy with a targeted therapy, in this case a focal adhesion kinase inhibitor, where we looked at these cell lines in 3D, and we gave irradiation alone, the focal adhesion kinase inhibitor alone, or the combination, and then we looked at the colony formation afterwards, and you can see that in both of these cell lines, you got a benefit of combining a targeted therapy with radiotherapy. So you can test your hypotheses in vitro using cell lines in 3D, but you can also apply this to patient-derived tumors. So here we also looked at these patient-derived breast cancer mammosphere colonies, where the focal adhesion kinase was expressed and activated. So this is phosphorylated focal adhesion kinase. And then we could use these using patient-derived breast cancer cells. And here we took 4 patient tumors and showed that the combination of irradiation and the targeted therapy was more efficacious than either alone. So these are the kind of preclinical in vitro models from patient samples that you can use. But I think now, over the last few years, we've become better and better at growing these patient-derived organoids, and in particular, many people have been using this method published 4 years ago by Sachs et al., where you take patient tumors and you derive these PDOs, or patient-derived organoids, and you grow these in 3D in Matrigel, and so you can look at patient-orientated responses to radiotherapy and targeted therapy using these. You can also take patient-derived xenografts that you've derived and put these into organoid culture, and this enables you to test therapies, and then you can go to the in vivo setting and validate that they're going to work in an in vivo setting. So I think these are very applicable preclinical models to use for combining targeted therapies with radiotherapy. So in terms of xenograft tumours in mice, we know that you can grow cell lines in these immune-deficient mice, but in fact, over the last few years, many people have grown these patient-derived xenograft tumours in the mice, and you can grow them in various places, subcutaneous, orthotopic in the mammary gland, and people have developed an intraductal model that seems to be more able to grow many more of the patient samples, and you can do metastatic modelling in these samples, but what you can't do is model the immune system since they're immune deficient. And for that you need immune competent models where you use mouse syngeneic tumours, but those don't really model human tumours quite so well. Some of them are better than others, I would say. So this just shows a schema by which we generated PDX. We took surgical samples, or we took ascites and pleural effusions from metastatic late breast cancer patients. And then you can use those both for 3D culture and for putting into mice. We use these most immune deficient mice, the so-called Nodsky gamma mice or NSG mice, and then you can passage these patient-derived tumors through the mice. And so you can generate experimental groups to do placebo control type experiments. These have worked out to be very good models of the patient, although you can't generate them from every patient. There's about probably a 20 to 30% success rate in these NSG mice, less so in some of the older mouse models. But what I can show you is that histologically, this shows you the primary tumor that they were derived from. This shows you the PDX, and they model histologically the original tumor. Also, they express, for example, the ER, the PR, and HER2 that was expressed in the clinical sample is expressed in the patient-derived xenograft in the mouse. And we formed this EuroPDX consortium, and we published 3 years ago now that those using over 1,000 PDX models from the consortium, that they maintain the genomics of the original tumor, and that's maintained across passage. So they are good models of the original patient tumor, although they do lack this immune component. So we've also showed, and many others have showed, that they will undergo spontaneous metastases to the lung and also to the bone, and you can see the percentage across these 6 different patient models. And so they can also be used to model those metastatic tissues, although there will be some differences between the tissues in a mouse and the human tissues, but I think they can be useful in guiding your clinical approaches. So just to summarize these preclinical models, I think I've shown you that the patient-derived samples are good for use in these in vitro 3D culture models, and in particular, using them in vivo, you can model individual patient response and also metastatic modeling, but you're less able to model the intact immune system. So I wanted to talk a little bit about the CDK4/6 inhibitors and what evidence there is for rational combination with radiotherapy. And to do that, I wanted to go over again some of the mechanism of action that was covered earlier and go a little bit more into the resistance mechanisms. But I think there are opportunities for rational combinations with radiation therapy. And as was shown earlier, I've picked out the Paloma-3 data, but there has been this evidence that palbociclib, ribociclib, and abemaciclib have benefit alongside endocrine therapy in advanced breast cancer, and also now more so in the adjuvant setting. And it was reviewed earlier, the potential interactions with radiation and toxicities in the clinical models, and I'm not really going to talk about toxicities, more about the efficacy of the CDK4/6 inhibitors and how their mechanism of action might support their rational combination with radiation therapy. So a couple of speakers have gone over this, but the cell cycle is obviously the target of CDK4/6 inhibitors, and this just shows a model of the cell cycle from mitosis through DNA replication back to mitosis. And it was really a former tutor of mine at university, actually, Paul Nurse, who discovered CDK1, which is this cyclin-dependent kinase here, and also other regulators such as WEE1. And he got a nice little prize for that 20 years after the discovery. But it's really the homologue of CDK1 in the G1 phase, this CDK4 and CDK6 that have been the targets of palbociclib, ribociclib, and abemaciclib. And as others have said, these kinases are responsible for this phosphorylation of RB, and that's a key step in the cell cycle. And so by inhibiting them, you're potentially inhibiting this RB phosphorylation and blocking the cell cycle here. So in our prior to this meeting, the group of preclinical faculty went through the data from the literature to try and find papers where they had tested interaction of CDK4/6 inhibitors and radiotherapy, and in fact, Elisabetta Marangoni went through these papers. And essentially, they're kind of ranked here, 4 papers. There wasn't a great deal of literature. And they're ranked here really in kind of what we thought to be their importance to a degree in their use of cell lines or human models or use of a multiple number of models. They gave various fractions of irradiation in combination with the CDK4/6 inhibitor. And our conclusions are here, and some of them lacked use of the models in vivo. They were just simply cell line studies, mainly in 2D. But I guess this top one was the most informative in a way, and concluded that basically if you deliver your radiotherapy before palbociclib, you actually can mediate a superior anti-tumor effect through the change in the tissue microenvironment after you administer the CDK4/6 inhibitor. What I wanted to focus on a little bit were the resistance mechanisms, because I think, when you're giving CDK4/6 inhibitors, not all patients are benefiting from them, and some patients benefit and later their tumor will progress. So really, there is resistance in almost all tumors to CDK4/6 inhibitors, depending at the stage that you give them. And those resistance mechanisms that have been studied have revealed several resistance mechanisms, but some of those are important because then you'll be able to sequence drugs that can target those resistance mechanisms. And if you're given radiotherapy at the same time, obviously you need to know that targeting the resistance mechanisms is also going to complement the radiotherapy. So these that I'm going to review are those that relate to the cell cycle, those that relate to other signaling pathways, but also the activation of the immune cells, which may be important and relevant to the use of immune checkpoint inhibitors. So firstly, many of the first papers that came out, particularly this early one from Nick Turner, pointed out that one of the major points of resistance are the amplification of other G1 cyclins or the loss of RB. So if you lose the RB that's normally phosphorylated, then obviously you're losing the mechanism of action of the CDK4/6 inhibition. It's also been published that upregulation of CDK2, another kinase, cyclin-dependent kinase in G1, then again you're getting over the need for CDK4 and 6 that are being targeted by the palbo. And there's some evidence that targeting CDK2 and CDK4/6 in combination can be beneficial. With Violeta Serra in Barcelona, we published this preclinical work last year where we looked at, I think, 40 patient-derived xenograft samples and showed that actually the resistance to ribociclib coincided with high P16 expression, again an inhibitor of the G1 kinases, and also the loss of RB. So these seem to be very common mechanisms in resistance in that part of the cell cycle where the drugs are active. But there are other mechanisms of resistance, and some of those were revealed when the Paloma 2 and 3 trials were analyzed by gene expression, and in particular resistance to palbociclib was associated with G2M checkpoint, so the other part of the cell cycle, but also mTOR signaling and the activation of immune cells. So these were in the gene expression profiles. These were areas that were associated with resistance. And so which part of the cell cycle does that mean? It means this area here in G2, and in particular there are some kinases such as aurora kinase and polo-like kinase, which are known to be active in that phase. And Elisabetta Marangoni published this nice paper 3 years ago showing that PLK1 is particularly a target in those resistant tumors. And she did that by growing patient-derived material in mice, treating with palbociclib, and here you see no growth. But in the one tumor that starts growing, if you then take that tumor out and you analyze its gene expression, you see the hallmark is actually an increase in the G2M checkpoint. And so what you can do then is reimplant those tumors into another mouse, show that they're still resistant to palbociclib, but then apply a polo-like kinase inhibitor called volasertib, and you can see here in orange that can then overcome the resistance. So by targeting a resistance mechanism, you can overcome the resistance. Other pathways that have been associated with resistance include the mTOR pathway, which I'm sure you'll be familiar with because it's the target of everolimus. And so that has also been noted as an important pathway of resistance. I won't go into that in detail. But the activation of immune cells may also be important. So you're giving this CDK4/6 inhibitor, you're activating the immune response in resistance, but of course radiation can be complemented by the activation of the immune cells. So this might be a rich vein to exploit in the future. This just shows you the signature of these palbociclib resistant cells, showing that the T cells and the macrophage are enhanced by the treatment in those that are resistant. So really, I think what we're saying is that in the resistance phase of CDK4/6 inhibitor treatment, maybe when the tumors start to progress again, actually now you've got new targets for inhibition, including the cell cycle targets, G1 and G2 kinases such as CDK2, CDK7, aurora kinase, and polo-like kinase. There may be some evidence for going into those with drugs that inhibit the mTOR pathway, everolimus that hits it directly, but also AKT inhibitors that might be upstream of mTOR. And then, the findings are that you have activated immune cells, so maybe immune checkpoint modulators might be important post-CDK4/6 inhibition and potentially in combination with radiation therapy. So I think, just to sum up in my last minute or so, there's evidence that these CDK4/6 inhibitors can induce apoptosis, senescence, inflammation, that they can prevent DNA repair and activate immune activity, and all of these may be beneficial to radiation treatment, but there may be a requirement for new combinations of radiation therapy with other targeted therapies, in the CDK4/6 inhibitor resistant tumours that are going to be progressing. And those include targeting polo-like kinase, perhaps PARP inhibition, we'll hear about more of that later, the immune checkpoint, mTOR AKT, and possibly other signaling pathways that I've not talked about today, but STAT3 and WIMP1 appear to be important too. I'm 2 seconds over time, yeah. Sorry about that. So just to summarize, I think I've told you today about the superiority of using patient-derived cells compared to cell lines, and that you can use those in three-dimensional assays, either colony assays or organoids, and then do in vitro screening. You can then transfer those for validation into xenograft tumors in mice, where you can study resistance, as Elisabeth's studies show. You can study spontaneous metastasis and model advanced cancer. For the CDK4/6 inhibitors, there is preclinical evidence of a benefit of combination with radiation therapy. But I think you've got to be aware when the tumors are resistant to CDK4/6 inhibitors and they're advancing, then there are mechanisms of resistance that offer new opportunities for rational combinations with radiation therapy. So, yeah, that's where I'd like to end, and I just want to thank the preclinical faculty from the ESRO consensus, which is myself, Elisabeth Marangoni, Claire Isaac, Andrea Morande, and also Sven Rottenberg. So thank you very much for listening.